My big question is, can your discovery one day be used to extend the human lifespan?
Well,
There may be a couple of people in the audience who may not exactly know what induced pluripotent stem cells are and why they're important, so please bear with me. I'll just go through a few slides just to make sure we're all on the same page to begin with. I'm going to go back to 1998 when scientists first derived human embryonic stem cells from fertility clinic embryos that were destined to be destroyed. Derivation process is essentially taking an embryo at the 4 to 5 day stage. This is about the time in which the fertility clinic would transfer this into a woman's uterus for pregnancy. But again, these are embryos that the couple has decided they're not going to use and they've decided to throw them away. At that stage, you can derive embryonic stem cells from the inner cell mass and grow them out. What's remarkable about these stem cells is that they can become any kind of human cell in the body, going down these 3 major lineages. They do have limitations, however. If you're getting embryonic stem cells from fertility clinic embryos, you have very little control over the genetic composition of these cells. You just basically have to get what you get. In 2005, there was quite a bit of discussion of maybe getting customized embryonic-like stem cells by using what we now call cloning technology. And the proposal was to take a skin biopsy from a patient of interest, somebody with a genetic disease of interest, get an egg donor to provide her eggs through hormonal induction, and without fertilizing the egg, suck out the nucleus of the unfertilized egg, replace that nucleus with the nucleus from the somatic cell donor, literally charge it up with electricity or chemicals to fool it into thinking that it became a fertilized egg, and then you get your customized embryonic stem cells from that. Now, this was, as you can imagine, very controversial because where are you going to get all those eggs? How many eggs would you need for this kind of work? Are you going to pay the women like they normally get paid from fertility clinics? Isn't this the same technique they use to clone Dolly the sheep? Can you make human cloned babies this way? Lots of controversy, and the field sort of stalled out at this point. At this point, the President George W. Bush Council on Bioethics came up with some proposals to come up with ethically derived embryonic-like stem cells, pluripotent stem cells. And one of them, they had many proposals, but one of the proposals, they said, would solve everyone's problems ethically. It would be to reprogram adult cells to behave like embryonic stem cells. Now, they said in 2005, this is the furthest away. This is the most pie-in-the-sky dream. Well, 1 year later, it was shown that it could be done in the mouse, and a year after that, it could be done in human. There was a study that was done at the Harvard Stem Cell Institute where researchers fused a skin cell with a human embryonic stem cell using private funds. And this entire enormous fused cell that has twice the number of chromosomes in its entirety started to behave like an embryonic stem cell. So there must be something about the embryonic stem cell that can teach the skin cell to do what it does. If you imagine the human DNA as an enormous keyboard, if it's a heart cell, it's playing the heart cell song. Those genes are activated. If it's a stem cell, it's playing the stem cell song. Those genes are activated. Can you teach the skin cell to play the heart cell, the stem cell song, without fusing them together? So Yamanaka did an incredible thing. He used viruses to insert into the DNA of skin cells to sort of try to teach the DNA to behave like an embryonic stem cell, and he narrowed it down to 4 factors that are extremely active in the stem cell state. These are now called the Yamanaka factors. This then allowed cloning without cloning, essentially. It allowed the dream of cloning for research without actually using cloning technology, because you could take an ALS patient, any patient of interest, get their skin cells, or really any cell from their body in some non-invasive way, reprogram them to make pluripotent stem cells that can then become any kind of cell in the human body. And this is extremely significant for lots of reasons. Basic biology, you can try to understand what goes wrong in the diseased patient versus the healthy control. At what point do things start to go awry in development? So you can compare. It also gives you an unlimited source of cells for drug screening. both for safety using healthy cells like liver cells, and also disease-targeted screening for disease cells. And these are just a couple of the many, many applications we'll be talking about tonight. As people say, the rest is history. In 2012, Shinya Yamanaka, along with John Gordon, got the Nobel Prize for Physiology or Medicine for the reprogramming of mature cells. I'm very excited now to bring out our guest. Would you please welcome Shinya Yamanaka? You were here because you ran the Boston Marathon a few days ago. How was that?
Yes. Well,
He walked here from there to here without limping.
well, running Boston has been my dream, so the dream came true. But it was very warm. I was sick last week.
All these excuses.
Yeah.
It was warm, it was sick,
I
it
was
was
still
uphill.
in jet lag, but I was still able to finish it. So, and I most importantly, I really enjoyed it. So, thank you
Great.
so much, Boston. Thank you.
Thank you. I've heard you say that doing science is like running a marathon. What do you mean by that?
Just like life, science is really a marathon. It takes long. Sometimes it goes well, but most of the time, like the day before yesterday, it doesn't go well. But nevertheless, you keep running. No pain, no gain, in order to succeed in marathon and also in science. So I've been learning a lot from marathon to be a scientist.
You and I first met in 2006. I'll never forget this. It was a very small meeting in Norway, at a very remote village in Norway. I don't know why we were meeting there. It was an invitation-only small meeting. I was there as a bioethicist. And you had been there, you arrived just after your mouse paper was published in Cell 2006. I remember at that meeting, there were a few people, a few scientists there that kept doubting what you did. One person said, "Are you sure your cell culture wasn't contaminated?" And I thought, well, what kind of contaminant could possibly be? I want to know what that contaminant is that would make them pluripotent. So did you face a lot of doubt when that paper first came out from other people?
Yeah, very much. So I attended another meeting in New York called Spring Harbor meeting. And after I gave a talk, they have a very nice pub. So many people were drinking. And when I attended, I was able to listen. Some people were talking, only 4 factors.
Yeah.
It shouldn't be true.
Uh-huh.
Then they identified me and they say, "Oh, Dr. Yamanaka, please come, please come." But I think it was very reasonable because even to us, it was surprising. Actually, when the first author, Kazu Takahashi, came to me, "Dr. Yamanaka, it looks like we succeeded in making ES-like cells from mouse skin cells. And I saw under microscope, the cells look like ES cells." But I told Kazu Takahashi, "Don't be excited. 99.9%, this must be something mistake. This must be probably contamination of ES cells. Because we have many, many mouse ES cells in our lab. We are very careful not to make any contamination. But ES cells are very tricky. Only one cell contamination is sufficient to make us in trouble." So I told him this must be some kind of mistake. And I asked him to repeat it. He repeated the same experiment again and again, but it always worked. But I was not convinced enough. So I have 2 or 3 more senior members in my lab. I asked them to repeat a very similar experiment, but in a little bit different way. And it all worked. So now we were convinced So that's how it happened to us.
Wow. Now, that was also right at the time that a Korean group had claimed that they had done the cloning approach and got patient-specific stem cell lines that way. After the publication of their work, I started working with them to sort of come up with ethics policies for their institute, and then it became exposed as being fraudulent. Now, that was a terrible time for the Korean teams because everybody that was on that paper were investigated, and there were many excellent young investigators that had their careers ruined. I noticed in your mouse paper there were just 2 authors. Were you influenced at all by what happened in Korea?
We were, yes, it was after that Korean scandal in Korea. We were very nervous. So publishing that first paper, usually publishing a good paper is a joy for
And it was
scientist.
a Cell, it was a great journal.
Yeah, it's a Cell. But for that particular paper, we were kind of afraid of the response we will receive from the world. Not only from scientists, but also from general, like media people. So I talked to Kazu and other potential authors. Maybe we should limit the number of authors as small as possible. So the first author, Kazu, who did the most important part of the work, and also myself as the corresponding author, to be listed as only 2 authors of that paper, because we were responsible. We were responsible to receive any criticism from scientists or from media people. So that's how we published that first paper.
Now, before the publication, before you got your positive results, was there any doubt? I mean, you were sort of doubtful when you got the result, but did anybody think, this is a crazy experiment? Why are we doing it this way?
Yeah. So we knew it's true because we have repeated many times in the lab. But once we submit the paper or once we publish the paper, we have to see if other scientists in other laboratories can reproduce our work. And it's going to be tricky because those stem cell works, like any small differences, like the quality of water can change the result. So that's one of the reasons why we were so nervous. We
you had good water in Kyoto.
Yeah, maybe we should ship all the water from Kyoto to Boston or to whatever is required. But we're very lucky or unlucky. Within the year, within like 6 months, people in Boston published another paper reproducing our experiment. So it was the beginning of fierce race. But at the same time, it was a huge relief on us. So it was a very strange moment. Yes.
I wonder, why did you start with the mouse skin cells? Why didn't you just start with human?
Well, it's a very good question because for many reasons, mouse is much easier than human cells. For example, or if you want to convert normal human cells to cancer cells, it's much more difficult than mouse cells. So in order to make mouse cancer cells, all you need is only 1 or 2 oncogenes. But in human cells, we know that we need at least 4 or 5 combination. So that was one reason why we thought we should start on mice.
Were you surprised that it was the same for Yamanaka factors that worked for human?
We are very surprised. Because of that cancer example, we thought in mice it was 4 factors, but we thought in human it would take probably 6 or 7 factors. So combination of 4 is complicated enough, but combination of 6 or 7 is even more complicated. So we thought, wow, I don't know how long it would take from mouse to move from mouse to human. But to our surprise, the same 4 factor worked on both mouse and human. So it didn't take that long. It took less than a year to move from mouse to human.
A few very key teams almost simultaneously published in different journals the human iPS cell work.
Exactly. Yes.
Jamie Thompson's lab in Wisconsin used Lin28. So they actually used one factor that was different. Did that surprise you?
It was surprising. Actually, out of 4 factors, they also used 4 factors. 2 are common. OCT4 and SOX2 are common. But the remaining 2 factors are totally different. So it was surprising.
Now, what were you doing when Stockholm called you?
It's a very good question. It was a holiday in Japan 12 years ago, 2012. I was at home. My wife here, she asked me to fix our washing machine. So I lay down and I was doing my best to fix our washing machine because otherwise my wife would upset. But all of a sudden my cell phone, yes, I got a cell phone call and it was in English. And he said, well, Dr. Yamanaka, we have decided to award you this year's Nobel Prize. And to my surprise, in the end, he asked me, would you accept? I was very surprised. And of course, I answered, yes, why not?
Why not? Did you finish fixing your washing machine?
Actually, I failed. But our government, Japanese government, was kind enough. They gave me enough money to buy a new washing machine.
That's great. I'm glad to hear.
Yes.
Now, how old were your daughters when this happened?
Oh, they got surprised, of course. But to my surprise, they maintained calmness. So yeah, it was it was very good response. I thought it might make them very unhappy, but they took it very naturally. So it was okay.
Wow. Now, it's funny because the award is the Nobel Prize for Physiology or Medicine. You pick. Now, your discovery is important for both. What are some of the applications of iPS cell technology today that you find very exciting?
So at that time, we are trying to use iPS cells in disease modeling and drug discovery, as you just explained. And of course, another very important application is cell therapy, to transplant like heart cells from iPS cells to patients suffering from heart failure. So those are the 2 major medical applications of iPS cells for patients. And we have been working very hard to realize those 2 medical applications.
Now, when you first made the discovery, were there any potential uses of iPS cell technology that you thought were a little bit concerning to you?
Yes, that's very true. In the beginning, in the first week after we succeeded, we published our first mouse paper. We are very happy because we thought we have overcome a big ethical issue about human ES cells, in that we don't have to use human embryos. So I thought, well, we have overcome a huge ethical issue. But after a week, I realized, wait a minute. We can now make like germ cells from iPS cells, from your skin cells, or from your blood cells. Well, probably we have generated even bigger ethical issue by making iPS cells. So it was a kind of shocking moment to me. I went to the Japanese government, Ministry of Education, and asked them to start an ethical conversation about that kind of ethical issue of iPS cells. And they quickly responded. And at that time, I took, at least in theory, we could make germ cells like sperm and eggs from iPS cells, but it was already 17 years ago. I thought it was just by theory. I thought it would take very long, 20 years, 30 years, before we can actually make germ cells from iPS cells. But it took much shorter than that. So within like 5 years, one of our colleagues in Kyoto University, they succeeded in making sperm and now even eggs from mouse iPS cells.
From male mice.
Yeah, now, yes, they can make eggs even from male iPS cells.
And they made mouse pups.
Yeah, exactly. And that doctor, Dr. Hayashi, is one of the Time 100 most important person this year. So it's moving very quickly. It's moving very quickly. So yes, we have generated new kind of ethical questions.
As a bioethicist, people would tell me when your discovery was made, "Oh, Dr. Hyun, I think your work is done now with stem cells because now they can make them from any cell." And I thought, just wait. What I thought was significant was for embryonic stem cells, the genetic source of the cell line is destroyed during derivation. There's no existing cell line donor. But with iPS cells, you could have a lifetime where in some cases, the pediatric patient has an iPS cell line and will grow up with this cell line in existence being shared all over the world. And so that could be concerning because now stem cell research has brought on the baggage of genetic research and privacy. So many other things to think about. What I find fascinating, I'm curious to know your thoughts, the animal work done with iPS cells and even conservation. We're talking a little bit about the sperm and eggs made from that. But there are people who are hoping to use iPS cells from endangered species to rescue them. Is that something that you find interesting?
Yeah, it's another very interesting and important potential of iPS cells to preserve those species in danger. And I understand some scientists, they even make like mammoth by using elephant iPS cells. So the potential is, in addition to those two major medical applications, which you and I just discussed, there are more potentials of this technology. So I have been just amazed.
Yeah, another area that's really exploded in stem cell research and also for the ethics of it is making little three-dimensional models of either organs called organoids or even whole embryo models that seem to replay the first few weeks of embryo development. Using iPS cells, using embryonic stem cells, it kind of starts to, in some way, kind of go back to the embryo. Even though initially it avoided embryo work, there's some who are really fascinated by, but also a little bit concerned that iPS cells, skin cells, can be transformed into pluripotent stem cells that then can be assembled together to replay the developmental tape of you when you were.
That's very true. In the last few years, many scientists in the world, they are trying to recapture human embryos by using iPS cells or ES cells, and it's moving very rapidly.
Yeah. So this series is called The Big Question, and I have a big question for you. My big question is, can your discovery one day be used to extend the human lifespan?
Well, so we are doing our best to expand our health span. I'm not sure about lifespan itself. If it's healthy, if we can live to 100 or 120, that would be great. But I cannot imagine to live like up to 200. So it may be okay with that particular individual, but how about the society as a whole? So I just want to focus on health.
Mm-hmm. Medical application.
Yes.
Yeah.
Mm-hmm.
Now, we were in Stockholm for a meeting for the International Society for Stem Cell Research in 2015. We'd gone running together that morning. But later that day, you don't know this, later that day, I went to the Nobel Prize Museum. Okay, so I walked in, and a bus of Japanese tourists came into the museum, and they went straight to the Yamanaka display. So I followed them, and they're all looking at it. And I said, oh, Dr. Yamanaka is here in Stockholm. They said, oh, yeah, Yamanaka here. And I said, no, he's nearby for a meeting. They didn't understand. I said, no, Yamanaka here. And so I showed them a selfie that we had taken. And I showed it to them, and they all started bowing to me. And I thought, no, not special. But you are a cultural icon, a cultural treasure in Japan and around the world. Have you been able to use that as an opportunity to sort of, bring up other people, like champion causes? Because that's a very unique platform, I think, for a scientist to have that cultural weight and love.
Yeah, well, Nobel Prize means probably a lot more in Japan than in US. Because here there are many. In Boston alone, I don't know how many Nobel laureates. But in Japan, we only have less than 30 in total in history. So yes, whenever I'm in Japan, many people can recognize my face. So I need to behave very well. But I come to Japan, I come to San Francisco every month. I stay 1 week in San Francisco and 3 weeks in Osaka, Japan. Whenever I'm in San Francisco, I can relax. I can do whatever I want. I don't do anything bad, to be honest. But
your wife is right
here.
Yeah,
my wife is there. Yes. But in Japan, yes, that's very true. But I try to use it to raise awareness, to tell general people the importance of science, and also to attract young students to science. So I do use that kind of status in order to promote, raise, social prestige of scientists as general. Because many young people in Japan, if we ask like 10-year-old boy or girl, they may say, I want to be a baseball player. I want to be a doctor, but only a few of them would answer, I want to be a scientist. So I want to change that kind of atmosphere back in my country.
Well, speaking of, you started off, you were a doctor. So what first inspired you to become a doctor?
So it was, I had many reasons, but probably the biggest reason was my father. He was a very healthy person, but when I was a junior high school student, he became sick, and there was no cure for his disease. So he became weaker and weaker, literally every day, every month. So that made me very interested in medicine. And also, my father wanted me to become a doctor. Maybe he thought I would be able to help him. So I listened to him, and I went to a medical school, and I became a doctor. At that time, there was no cure yet for my father. And unfortunately, he passed away soon after I became a doctor. So it was very shocking because I couldn't do anything for my own father and also for many other patients. as a young doctor. I felt, powerless. So that was why I thought I should become a scientist, because it is science that could help those patients suffering from intractable diseases, not now, but in the future. So because of my father, I became a doctor, and because of my father, I became a scientist. I owe him a lot.
Right. When we were in Norway, we were struck by all these troll dolls everywhere. And I asked our host, whose name was Thor, by the way, what's the deal with all the trolls? And he's a medical historian. He said what he thinks happened was Norway was a seafaring nation. They would build the big ships using the timber of the mountains and then go out to sea on the fjords. And he thought that many of the sailors came back with syphilis. And so what you're seeing in the doll mythology are basically sailors who went crazy, living in the woods and under bridges and had strange bumps on their faces, which you still see in the troll dolls. He thinks that's probably where it came from. So a lot of times when people don't understand something, they build a mythology around it. Do you think there's mythology that's kind of building around iPS cells and what they can do? Do you think there's a little bit of a danger of people kind of filling in their lack of knowledge of the technology with some other story?
Yeah, that's one way we want to use iPS cells. Mm-hmm. So yes, that's actually I have been being amazed how many applications are there about stem cells and about iPS cells. So I really hope many scientists would use iPS cells in many ways that I cannot even think of.
Yeah, I think one of the things that has come up, some patients are very desperate for a cure, and they may hope that a clinic can give them their own iPS cells back because they're genetically matched to you. Most people believe it won't be rejected by your body. So there may be some unrealistic expectations about the technology. Were there unrealistic expectations about cures and when you became very well known in Japan, and did you feel some of that pressure?
Yes, it's a very important point. It's many. So there are many types of stem cells. iPS cells, some somatic stem cells like bone marrow stem cells, but some kind of unproven so-called stem cells. And for many patients, they do not distinguish those stem cells. So it is very important to explain very well what's the difference between like iPS cells and some kind of so-called stem cells. And we need to tell them, well, we are working very hard day by day, but it takes some time, just like marathon. We cannot finish it in 1 minute. It takes years. It takes 5 years, 10 years, or even 20 years. So that's what I have been trying to do, trying to explain as much as possible in a very understandable way. But I found it actually very challenging. Yes, so you must be much better in doing that. So
I try, but this job also helps a little bit. So you're celebrated for your great successes. You must have had some failures. Like, give me an example of a scientific failure that you experienced.
Well, I'm making failures every day because science is full of failure. But in my very first experiment as a young postdoc, a graduate student, it was almost, 35 years ago. I did a very simple experiment, but the outcome was something totally unexpected. It was very different from what I and my mentor expected. So it was kind of a big failure. But I was very excited. Instead of being disappointed by that unexpected result, by that kind of failure, I got very, very excited by the unexpected outcome. I was more surprised by my own response than by the unexpected result itself. So that was the time I realized I should become a scientist instead of becoming a doctor. So that first failure or unexpected result played a very important role in my life and also to making iPS cells. So in a sense, I was very lucky. Yeah. The first experiment did not go well. Yeah.
Yeah, I have one more question before I turn to questions from the audience. Do you think there's an important role for ethics in iPS cell research?
Yes. Many aspects. Once again, we can generate germ cells. We can generate brain organoids. We can generate human embryo models. And we can generate like human organs in pigs by using iPS cells. So the question is how much we can do, how much we should do. We may be able to help many patients, but it's just like any other science. It's really a double-edged sword. If somebody tend to use it in a wrong way, we can make the world worse place. So ethics is really, really important. As a matter of fact, in our institute in Kyoto, we have a group of bioethicists headed by Misao Fujita, who must be a good friend. So I think it's very important to work together, the scientists and bioethicists work together in the same building, day by day. So I think ethics is really, really important.
Yeah, I knew that you were very, very interested and sensitive to ethical issues from the very beginning. So when your mouse paper came out, George W. Bush vetoed for the second time a congressional legislation that would have allowed for more federal funding for human embryonic stem cell research. And when he vetoed it, he said there's research coming out of Japan that you can make stem cells out of skin cells. He didn't say in the mouse, but he said, you could do that. So he's more committed to vetoing this. And I told you about that. We were in a meeting in Australia. I told you that George W. Bush just said that. And so we decided to write a commentary together where you have to, along with you, Rudolf Janich at MIT, Konrad Hochlinger at Harvard, the four of us wrote a commentary that said, just because you have iPS cell technology doesn't mean that you can stop human embryonic stem cell research. They have to develop together, and you have to compare the two. And it was for scientific and ethical reasons. There's a need to sort of keep, for safety reasons, to keep both areas of research active. So you were a part of that, and that was actually one of the most cited papers that year. And then shortly after that, the human work started to come out. And so I agree. I mean, I think that the approach of going forward in these new frontier areas with ethics and science is extremely important, and they have to be intertwined. I'm going to turn to questions now. I'm sure many have come in. What were some of the best and worst advice you received in your career? Best and worst.
Best and so the best advice was from my mentor. And when I got the unexpected result, when I was a graduate student, and also when I was a postdoc, I got very unexpected result. And in each cases, my mentor, they encouraged me to continue working on that unexpected results. Because of their encouragement, I was able to continue my research, which actually eventually resulted in the generation of iPS cells. So that was the best advice. Worst advice, it's very difficult. But probably when we started this project, trying to generate iPS cells, many many of my colleagues, they try to talk out of this project. Dr. Yamanaka, this is too risky. You should stop this. So that may have been the worst advice, but very reasonable advice. So I thought I should encourage young people, young scientists, to take risks, because I learned from my own experience. But now I am very old, and I need to teach many young scientists, and I'm responsible for many young scientists for their future. I ended up trying to stop their risky project. So it's much easier said than done.
Many people want to know, could you tell a little bit more the story behind how you narrowed it down to just those 4 factors?
Okay, we initially had 24 candidates. And when my colleague Kazu, he mixed all the 24 factors and put them all together into mouse fibroblasts, and it worked. We observed emergence of some ES-cell-like cell population. So then we need to narrow down from 24 to whatever it required. Well, we had no idea how many out of 24 were required. So it was a very difficult task. But once again, Kazu did a very nice experiment. Instead of testing all the combination of 2, combination of 3, combination of 4, combination of 5, it's an enormous number. He just took one factor out of 24 and tried the remaining 23 factors. So only 24 combination. And just one experiment, we found out those 4 factors are essential, because when he removed one of those 4 factors, the remaining 23 factors could not do the job. So it was a very smart but very nice experiment that he did.
Here's a good question. Have you ever lost your motivation to do research? And if so, how did you get your motivation back? I think this is coming from a young researcher. Because there are no old researchers that lost motivation.
So when I was a student or a postdoc, I mainly worked by myself. At that time, it was very difficult because, once again, science is full of failure. Almost every day, the result is not good. And I was working by myself mainly. So at that time, it was kind of difficult to maintain my motivation. But once we started working as a group, it was much easier because we can talk to each other, we can help with each other. So working as a team is probably important to maintain motivation. And now, after reporting iPS cells, not only our colleagues, but we have an opportunity to meet many patients or family members of patients. They are our huge motivation every day. And they are amazing. We know because the kids are suffering from intractable diseases. So each day they're having a hard time. But they often tells us, Dr. Yamanaka, please take care of yourself. So it's really moving. I know they're having a hard time, but instead of taking care of themselves, they are worrying about our health. They say, don't work too hard. So those conversations with patients and their family members is the biggest motivation right now.
Do you see differences in how your area of research is conducted in Japan versus the US? In terms of science itself,
it's very similar, but the funding is very different. In Japan, we rely heavily on the government, funding from the Japanese government. But here, of course, funding from NIH is important, but we have many other sources, funding from each state, and more importantly, funding from private sectors, fundraising. So I think the situation here is much better than in Japan.
What do you think is the next big thing in stem cell research?
So I hope many applications, more than 10 applications using iPS cells are now in clinical trials. So I really hope in the next 5 years or 10 years, many of them will go more general, will be approved by the government, by FDA or PMDA in Japan, so that many patients can have those new therapies. So that's the biggest hope right now.
Can iPS cell research cure baldness? I'm asking for a friend.
Well, it's very important for me and probably for you. But it's not life-threatening. So we are not working.
I think you're going to get a lot of volunteers for that clinical trial. That's the thing.
But many groups are working on that.
Yeah,
actually, and they're making very surprising progresses. So I'm hoping
there might be a Nobel Prize in that one too. Last question here: what do you think are important qualities to be a good scientist? What are the important qualities?
Okay, I would say curiosity. I think that's the number one. It's not your score at school. And whether you can enjoy failures, whether you can learn from failures, I think it's very important to be a scientist.
Well, I'm going to close this out by just recalling my favorite memory that I have with you. So it was back to that original meeting in Norway. I come down in the morning to go for a run, and you were stretching in the lobby. And you said, you want to go for a run together? So we went for a run. We were in completely unfamiliar territory. It was beautiful, but the fog was coming off the ground. We were in the woods. And we didn't know where we were. We were deciding, do we go left? Do we go right? Where should we go next? And we're running together. And I thought, this is ideal, science and ethics going together and exploring something new. Thank you so much for joining us here today. It was a pleasure. Everyone, Shinya Yamanaka.
Thank you. It was fun. Thank you very much.